Welcome back to the OmicsLogic Bioinformatics for Precision Medicine program. In this video, we will be reviewing what we have learned in session 2. The second session was designed to take you one step further by starting your journey towards genomic data analysis for precision medicine. After completing the session video and associated resources, you will know what is genomic data, what does it contain, how is it organized, and what can we find that is relevant for precision medicine. To briefly review what we have learned in session 2, first, we laid the foundational knowledge for genomics and covered topics such as what is DNA code, what do we already know about it, and what kind of answers can we find there. Then, we learned about the Human Genome Project, which is an ambitious research effort aimed at determining the DNA sequence and the location of the estimated 100,000 human genes. Further, we learned about the difference between genetic variation versus mutation, a specific type of variation that can lead to harmful results. For example, a change in function that is harmful is called a mutation. Next, we learned that when analyzing the genomic data, we are looking to find and characterize the changes in the DNA code and associate them with phenotype. We also discussed about some important acronyms to remember about genomic variations such as SNV, SNP, and CNV. Next, we learned about variant calling and annotation. Here we learned that during DNA variant calling, the analysis is performed with FASTQ files and that there are several important steps to consider, such as pre-processing, mapping, variant calling, and annotation. For this purpose, we discussed about the demo pipeline called the breast cancer mutation oncogene variant calling for TP53 gene on T-bioinfo server, and also discussed briefly about FASTQ, BAM, SAM, and BCF files. We also learned about how to visualize the resulting output on JBrowse. We also learned about variant call format that specifies the format of a text file used in bioinformatics for storing gene sequence variation. Now that we have learned about the difference between mutations and variation and how to identify these variations, in the next part, now we will learn about the clinical relevance of mutations. Upon receiving the DNA sequencing results for a sick patient, the first question that is usually asked is, what genes are affected? There is not a simple answer to this question. Many of the DNA variants are still being discovered, annotated, and rigorously analyzed by a diverse team of scientists. For example, in cancer, the DNA variation can help determine genetic risk associated with oncogenes, but most often, oncologists look for somatic mutations in tumor suppressor genes, oncogenes, or DNA repair genes. Since cancer most often occurs with age, many researchers see the accumulation of somatic mutations in key genomic regions, such as a combination of driver and passenger mutations that can help diagnose cancer early on, evaluate risk of cancer severity, and select appropriate treatment to slow down incurable metastatic cancer. Next, we learned about ClinVar, which is a freely accessible public archive of reports of the relationships among human variations and phenotypes with supporting evidence. Another great repository to annotate variants for cancer is Coursework, which stands for the Catalog of Somatic Mutations in Cancer. Here, one can estimate the clinical relevance of the variants by comparing it with a list of mutations compiled in Coursework database. Further, we also briefly discussed about loading VCF file in a data analysis environment like R for visualization, manipulation, and analysis to be performed on the entire dataset at once. To continue learning, we invite you to explore the associated resources for session 2, which comprises of coursework, case studies, and other examples from literature. The contents of session 2 correspond with practical lessons that you can complete independently, available on the OmicsLogic Learn portal. For session 2, we have covered concepts based on lesson from Course 3 Genomics. This course serves as an introduction to the bioinformatics subdiscipline of genomics, where students will be familiarized with the biology of genetics and genetic variation while considering practical applications. Within the full course, specific lessons that are of relevance to session 2 include Lesson 3, Introduction to Genomics, that will give you an overview of genetics and genetic variation. Lesson 5, Genomic Variation in NGS Data Practical, where you will learn to create variant calling pipeline to identify variants between cancerous and non-cancerous genome. Another lesson is Lesson 9, Advanced Genomics Overview, that will cover advanced concepts in genomics, including single nucleotide variants, various terminologies associated with mutations, and file formats that store variants data. Another lesson is lesson 10, analysis of NGS genomic data. Here you will analyze genomic data and interpret biological insights using JBrowse. The full course contains a lot more information that you can choose to explore independently at your own pace, so do not worry about completing all of them immediately. Another great resource that you will have access to is Project 14, Breast Cancer Oncogene Variant Calling. Here you will find answers to questions such as, what are these harmful mutations? What is their nature? To what extent are these mutations responsible for causing diseases or disorders? How can these prominent mutations be helpful to improve prognostication? In the upcoming session 3 on phylogenetic analysis and multiple sequence alignment, you will build upon the genomic data analysis skills that you learned in session 2. So if you have not gone through session 2 yet, make sure to complete the associated coursework and example projects listed before proceeding to the next session. In session 3, you will learn about data preparation for performing phylogenetic analysis and multiple sequence alignment and biological interpretation of the outputs. You will also learn about biological implications of identifying variants of concern, topics such as genome-wide association studies, and development of multi-drug resistance in a pathogen infecting a single host are also discussed.